A warm welcome to today's session on bioenergetics. Myself, Dr. Mausmi Shankar Adala, working as an assistant professor in the Department of Biochemistry, SGCWA. Learning outcomes will be on bioenergetics basics like exothermic, endothermic reactions and thermodynamic principles. It is a branch of biochemistry that focuses on how cells transform energy, often by producing, storing or consuming adenosine triphosphate, which is abbreviated as ATP. Bioenergetic processes such as cellular respiration or photosynthesis are essential to most aspects of cellular metabolism, therefore to life itself. Three types of fields in bioenergetics are respiration, photosynthesis and metabolism. Principle of bioenergetics is that the total energy of any system is always constant. It can be changed from one form to the other but can never be created nor destroyed. Free energy is the energy available to do work. Bioenergetic processes include glycogenesis, gluconeogenesis and citric acid cycle. What is enthalpy? Enthalpy is a measurement of energy in a thermodynamic system. The quantity of enthalpy equals to the total content of heat of a system, equivalent to the system's internal energy plus the product of volume and pressure. The SI unit for specific enthalpy is joules per kilogram. It is always indicated as delta H. Entropy. Entropy is a measurement of a system's thermal energy per unit temperature that is unavailable for doing useful work. Because work is obtained from ordered molecular motion, the amount of entropy is also measured of molecular disorderness or randomness of a system. The SI unit of entropy is joules per mole or joules per kilogram. It is indicated as delta G. Exothermic reactions is a chemical reaction that releases energy by light or heat. It is indicated by negative sign. Coming to the endothermic reaction, an endothermic reaction is any chemical reaction that absorbs heat from its environment and it is indicated by positive. Free energy of a reaction. The free energy change, that is the delta G of a reaction, determines its spontaneity. A reaction is spontaneous if delta G is negative. It is indicated by the following reaction where delta G is given as delta G naught plus RT integration of products by reactants. The delta G of a reaction may be positive and the delta G may be negative depending on the cellular concentrations of the reactants and products. Many reactions for which the delta G is positive are spontaneous because other reactions causes depletion of the products. Maintenance of high substrate concentration. At equilibrium, delta G is 0. So the equilibrium constant, that is a K equilibrium constant, which is indicated by the K form, also becomes constant, and usually the parameters used are temperature 25 degrees centigrade at pH 7. So these are the different values of the equilibrium constant with respect to the delta G at various spontaneity of energy coupling. A spontaneous reaction mixture may drive a non-spontaneous reaction with a free change of coupled reaction as additive. It is of two types. In the first case, some enzyme catalyzed reactions are interpretable as two coupled half reactions, one spontaneous and the other non-spontaneous. At the enzyme active site, the coupled reaction is kinetically facilitated, while individual half reactions are prevented. Free energy changes of the half reactions may be summed to yield the free energy of the coupled reaction. For example, in the reaction catalyzed by the glycolysis enzyme hexokinase, the half reactions are ATP plus water gives ADP plus inorganic phosphate, and in the coupled reactions, the ATP plus glucose can be coupled with the inorganic phosphate plus glucose reaction also. The structure of the enzyme active site from which water is excluded prevents the individual hydrolytic reactions while favoring the coupled. In the second case, two separate reactions occurring in the same cellular compartment, one spontaneous and the other not, may be coupled by a common intermediate. A hypothetical but typical example involving pyrophosphate. In the enzyme 1 catalyzed, A plus ATP gives B plus AMP. In the second enzyme catalyzed reaction, pyrophosphate plus water gives 2 inorganic phosphates. Overall spontaneous reactions may be A plus ATP plus water gives B plus AMP plus 2 inorganic phosphates. Pyrophosphate is often the product of a reaction that needs a driving force. Its spontaneous hydrolysis, catalyzed by the pyrophosphate enzyme, drives the reaction for which the pyrophosphate is a product. Coming to the enzyme coupling reactions in the transport, the ion transport may be coupled to a chemical reaction, which is the hydrolysis of the synthesis of ATP. In the diagram, it was not shown clearly about the involvement of this hydrolysis, but it should be recalled that the ATP hydrolysis or the synthesis reaction is nothing but water dependent, that is a hydrolytic reaction. The free energy change associated with the transport of an ion S across the membrane is indicated below. Since free energy changes are additive, the spontaneous direction for the coupled reaction will depend on relative magnitudes of delta G for ion flux and delta G for chemical reaction. There are two examples, active transport and ATP synthesis. The active transport spontaneous ATP synthesis is coupled to ion influx against a gradient. ATP synthesis spontaneous proton flux is coupled to ATP synthesis. What is oxidation and reduction? The evolution of photosynthesis and the generation of the oxygen that is now plentiful in our environment allowed development of metabolic pathways that derive energy from transfer of electrons from various reductants ultimately to the molecular oxygen. Oxidation of an ion atom involves loss of electron, whereas since the electrons in a CO bond are associated more with the oxygen, increased oxidation of a carbon atom means increased number of CO bonds. Oxidation of carbon is spontaneous, where two important electron carriers, metabolisms NAD plus and FAD plus, are produced. What is NAD plus or NADH? It is nicotinamide adenine dinucleotide. It is an electron acceptor in the catabolic pathways. The nicotinamide ring derived from the vitamin niacin accepts 2 electrons and 1 proton in going on a reduced state, forming NADH. NAD plus or NADH is similar except for the inorganic phosphate. NADPH is an electron donor in synthetic pathways. The electron transfer reaction may be summarized as NAD plus 2 electrons plus protons. It also may be written as NAD plus 2 electrons plus 2 protons. These are the redox enzymes associated with various proteins. There are an electron carriers that transfers electrons from glycolysis and Krebs cycle of the respiration to ETC of respiration. One NADH generates 3 ATP. Difference between ATP and NADPH is that ATP is a high energy molecule that stores and transports energy within the cells. NADH is a high energy electron carrier used to transport the electrons generated in glycolysis Krebs cycle to the electron transport chain. Nicotinamide adenine dinucleotide consists of 2 nucleosides joined by the pyrophosphate. The nucleosides each contain a ribose ring, one with adenine attached to the first carbon atom at first position and the other with the nicotinamide at this position. The compound accepts or donates the equivalents of ions. The functions. Nicotinamide adenine dinucleotide has several essential roles in metabolism. It acts as a coenzyme in redox reactions, as a donor of ADP ribose moieties in ADP ribosylation reactions, as a precursor of the second messenger molecule cyclic ADP ribose, as well as acting as a substrate for bacterial DNA ligases and a group of enzymes called cyretins that use NAD plus to remove acetyl groups from proteins. In addition to these metabolic functions, it can be released from cells spontaneously and by regulated mechanisms and can therefore have important extracellular roles. The enzymes that make and use NAD plus and NADH are important in both pharmacology and treatments for disease, drug design and drug development by designing enzyme inhibitors or activators based on its structure that change the activity of NAD dependent enzymes and by trying to inhibit NAD plus biosynthesis. Coming to the next important molecule, FAD plus and FADH. FAD means flavin adenine dinucleotide. It is derived from the vitamin riboflavin. It functions as an electron acceptor. The dimethyl oxazine ring undergoes reduction or oxidation. FAD accepts 2 electrons and 2 protons in going to the reduction state. FAD plus 2 electrons plus 2 protons gives FADH2. Given below is the pictorial representation of the FAD and FADH2 molecule. Flavin adenine dinucleotide consists of 2 portions, the adenine nucleotide and the flavin mononucleotide, which is bridged together through their phosphate groups. Adenine is bound to a cyclic ribose at the first carbon, while phosphate is bound to the ribose at the 5' carbon to form the adenine dinucleotide. It plays a very major role as an enzyme cofactor along with the flavin mononucleotide. Another molecule originating from riboflavin. Bacteria, fungi and plants can produce riboflavin, but other eukaryotes such as humans have lost the ability to make it. Therefore, humans must obtain riboflavin, also known as vitamin B2, from dietary sources. In biological systems, FAD acts as an acceptor of H plus and electrons in its fully oxidized form, and acceptor or donor in the FADH form, and a donor in the reduced FADH2 form. The functions: 1 FAD produces 2 ATP. FAD has a more positive reduction potential than NAD plus and is a very strong oxidizing agent. The cell utilizes this in many energetically difficult oxidation reactions such as dehydrogenation of its carbon-carbon bond to an alkene. FAD-dependent proteins function in a very large variety of metabolic pathways including electron transport, DNA repair, nucleotide biosynthesis, beta oxidation of fatty acids, amino acid catabolism. One well-known reaction is part of the citric acid cycle, which is also known as TCA or Krebs cycle. Succinate dehydrogenase requires covalently bound FAD to catalyze the oxidation of